the bioinformatics. To be very honest, let me share my thoughts before I start my own PhD, my own presentation. While I did my PhD, while I started my PhD, I was having no experience of computer whatsoever. That was the first time I started looking at computer as a system, that what exactly it is, and I have no idea how to work on the kind of a Unix system. So the first real bottleneck which the bioinformatics student face is basically what to do and what kind of system they should use. So my first word before I start anything start talking about bioinformatics, I would say, if you really want to be in terms of bioinformatics, want to really think in terms of bioinformatics, you have to change the way you are developing your own expertise in different form. I'm not saying that you should go and start learn computer, but you have to be passionate about what you want to do. Being a biologist, it's a different arena, but if you are well versed with whatever the tools in your hand, so then you are able to do anything of any sort. It's just like being a carpenter. Carpenter has all sorts of tools, but what he makes, that becomes a real gem. That's the real art comes up when they use those tools to do something very beautiful, very natural, and very different. And this exactly happens with the tools which is there in bioinformatics. There are a lot of tools, there are a lot of softwares, there are a lot of databases. We'll talk about that. But the real thing is, what you exactly want to do is to frame up the question. If your question is correct, and if your research question is real and worth working for, then these tools are helpful. But if you don't have any question, there's nothing to start with. So I would request all the students who are listening to me through this platform is to think and start believing that what are the exact questions we should ask for. Because a research question, for any kind of research, questioning is very, very important. And after that, you start looking for the ways to solve that particular question. Okay, so let me share my thoughts and the things which I want to convey over here. Just a moment. Okay, when Renu madam requested me that I should give a talk, and I was totally confused. What should I give? Because even I don't know, and I'm not convinced that bioinformatics is in the real shape exactly what we are looking at as a bioinformatics. But what is bioinformatics? Yeah, please go ahead. Yeah, is there any question? Something like that? Okay, let me go ahead with that. So the thing is that what kind of bioinformatics we are looking at and what kind of subject bioinformatics include. There is always a school of thoughts which says that bioinformatics is this, bioinformatics is that. But what exactly is bioinformatics? So I would say bioinformatics is a never-ending subject. There is no one book which can justify the bioinformatics. There is no one story which can justify bioinformatics. There is no one concept which can justify and say that this is bioinformatics. So my lecture will be orienting towards what exactly it was earlier or 10 years back, and now what exactly it has become. So that's why the title which actually Renu inspired in me was the evolution of bioinformatics. How do you really work out that bioinformatics and what kind of concept I should be needing to evolve everything? To be very honest, it starts from very basic. And what exactly are those basics? Let's look at those and let's go ahead with a discussion. Maybe I can keep last half an hour or 45 or 20 minutes for discussion. I will try to tell you the concepts. I'll try to take you to some of the websites which can give all you the aspects that how you can do such any kind of experiments. But as I said, that you have to frame up the right question. And what are those questions and how those would be framed can only be done when you start behaving like a researcher, a scientist. Because these are the questions, because some people ask me what bioinformatics gives you. But I always say bioinformatics gives me the hypothesis. And that hypothesis helps me to drive the science. So this is what exactly it is. And so how it is working. So let's look at that. The question is, why bioinformatics? If we say bioinformatics, so we clearly say that biologists are the bench studying small network of genes. So they are very happy. They were very happy at one point of time with one gene, one kind of scenario, characterize a gene, find out a function, and they're very happy about it. But bioinformatics has changed everything. And to be very honest, bioinformatics is not a new subject. Bioinformatics is happening right at the time when biologists were working at various levels. It even dated back in when it started with the Watson and Crick, and even before that. Why and how I'm saying so? I'm saying so because bioinformatics, when you do any kind of analysis, even if it is a bacterial growth curve doing in a microbiology lab, you use your mathematics skills, your computational skills to plot those graphs. And people were using various graph sheets to plot and then see what is the bacterial growth and how this can be optimized and all those things. So those are the bioinformatics. That was the starting of bioinformatics. It was not new, but yes, there are some canvases which changed over the time, and they have given a new way of looking at biology, completely different way of looking biology. So bioinformatics, as we say, the development and application of computational tools, which is say, and these tools are basically really tools. Why I'm saying so? This is because there are multiple aspects of data which we are producing through various biological experiments. Those are acquire, store, organize, archive, analyze, and visualize. So you can see there are multiple terms, and every term defines its own arena, its own workout, and its own fields. So you cannot say bioinformatics is one particular book. So you cannot say that. Acquire, that how you acquire the data, because data standardization format, when I show you some websites, you will realize that even these such kind of data you have for acquiring, there is a specific formats. Store, you design the database in such a way that it could be analyzed and given it to the researcher. Organized so that it could be analyzed in a proper fashion and do a comparative analysis. I'll show you how comparative analysis is done. Archive, that means the data which has been produced would always be optimized, would always be checked, would always be redone by various researchers across the world. So there is always a way that you will improve on that data. And then finally, visualize and analyze. When you visualize, you conclude some biological theories, a biological hypothesis. And that's why this is called as one of the biology's fastest growing subject or technology. And this is how we define bioinformatics. But there is a question and there is a problem. And the question is, how do you define bioinformatics? And some people say computational biology. So what is the difference between the two? And there is always a debate, because some people say that I am computational biologist, and other people say I am a bioinformatics person. But really there's a need to understand what is bioinformatics and what is computational biology. So bioinformatics is basically the biological problem posed by assessment and study of biodata. That means you produce any experiment, you do any experiment, you have some sort of outcome, you analyze that, and that comprises of bioinformatics. But the computational biology is basically the solution of the problems which arise due to bioinformatics. That means there are certain questions which need to be addressed because the biodata gave you some kind of hypothesis, and that hypothesis need to be sorted out, and those would be sorted out with the help of various tools and techniques, and those tools and techniques will be helping in developing computational biology aspect of it. So the bioinformatics professional develop algorithms, program code, analytical models, and all those kind of things. But computational biologist uses those kind of tools to address a specific biological question. And therefore, every bioinformatics question will be solved or be analyzed through computational biology. So if we say in a broader sense, what are the application of those two areas? If you see, there are numbers of lists which are listed over here. Now, if you see drug development, gene therapy, evolutionary studies, biotechnology, waste clean management, crop improvement, insect resistance, all those becomes the arena of bioinformatics because you devise algorithms, you devise various notional software workouts, you devise databases for bioinformatics. But when it comes to analysis of those subjects, let's say you have a bunch of data in your databases, then you go to these databases, look them for, and try to work out some kind of real-time simulations to see how exactly biological systems are working. So that's what stochastic models, systems biology, machine learning, molecular medicine, preventive medicine, artificial intelligence, all these comprises of computational biology. So those are the applications of computational biology. So in broader terms, when we divide among the subjects, so bioinformatics has got a lot of things. Even the computational biology becomes the subfields of bioinformatics. So various fields like genomics, genetics, proteomics, metabogenomics, transcriptomics, everything comes under bioinformatics. While computational biology, where bioinformatics becomes its subfield, where computational biomechanics, computational bioimaging, mathematical biology, theoretical biology, all those comes under the computational biology because you analyze the multiple types of data and try to produce some kind of hypothesis. And this is how you do computational biology. So computational biology is largely in the analysis mode, but bioinformatics is largely in the algorithmic and development of the tool mode. So these are two different arenas. So the student who is learning this subject have to define in which area I am good at. If somebody is very good at analysis, so those can be by computational biologists. But those are very good who can devise algorithms, understand the computational aspects very well, they can be a tool maker. So those are the bioinformaticians. So you can easily devise what are the demarcations which we can make. But the word of cautions, there is no division. But the point is, while you become the computational person and devise algorithm, you have to see the basic aspect of biology to frame up the question. So the upper hand is of biology, not of computation. So don't get confused. If you are a biologist, biology is your strength, and that would help you to devise research questions. And those questions will become the framework of your further research. So therefore, you devise the tools to do that kind of research. Then this is how you should space yourself, you should plan yourself and how you're going to do it. So basically how it is done. So you produce a lot of molecular data, and that is by the experimental mode, sequence, structure, functions, evolution, pathways, interaction, everything. And then you take that particular data, hypothesize something, and you take an approach to devise that I'll be doing systems biology to understand some nuances of complex how machinery of genes and proteins work. So you devise some kind of circuitry, you devise some biological systems, just like you devise an electronics person devise a circuitry. And this is how you become and develop an approach. Systems biology, synthetic biology, those become your subjects. So bioinformatics has evolved into some kind of approaches now. So more or less, bioinformatics is not stuck whatever we are. Now it has become something else. And let's look at that, how it became. So this was bioinformatics, which were 10 years ago, I should say. I have listed some softwares, some databases which are used. So bioinformatics were majorly divided into 4 different segments. There were databases, there were sequences, there were structure, there were pathways. When I talk about databases, there was a time when everybody was devising databases, new kind of databases, primary database, secondary database, and all those biological information which is coming through were putting in those databases. So that was the complete different world where people were focusing on creating that kind of data. Some people do some analysis from the primary data, make any database, something, a lot of databases were there, and a lot of databases were made at that particular time. Then came a time when we started talking about sequences, because those sequences of the biological data, we started putting in these databases and we started analyzing those sequences. That was a time when a lot of sequence search started coming up. BLAST, FASTA, HMM-ER, PyMol, muscle, multiple sequence alignment, and all those visualization, lot of things. Even the phylogeny came at that moment. So sequence becomes the basis of analysis, and you devise some novel hypothesis through that. And once this sequence and phylogeny were convinced, and people are convinced, they started going to structures. And how the structure field came up, that didn't came as a field, that came as an assistive workout, because X-ray was already there. NMR started quite later, but X-ray was there, and people were devising structures of various small proteins. And people started using the principles of physics and chemistry to do structural biology. And a lot of softwares, algorithms came up for visualization, for docking, for prediction, for simulation, everything started coming out. And when these two combined, people started talking about the holistic approach. And holistic approach is all about pathways. So databases and analysis started coming up. So people were not talking about one gene. People were not talking about one protein. People were talking about the holistic way. What is happening in the pathway? And what should I do? And what should be done? So that becomes the question. So these were the part, at least 10 years ago, bioinformatics was largely looking in this spectrum. So people were either working in databases, either in analysis of sequences, or analysis of structures, or analysis of pathways. But what happened? People started realizing that it's not about one gene or one protein, it's all about the whole genome. We talk about DNA. When we talk about DNA, there are large number of genes which is there. And those genes which we know might be expressing, might become or might not become an RNA. And then there is a RNA. There is always a question whether this RNA would translate into protein or not. If yes, whether this mRNA is really giving some meaning to the whole workout or not. Then these proteins became lesser than what exactly the RNAs were. And every protein will not participate in every pathways. So they might be just a kind of a signal protein. So that leads to some kind of metabolite. That means every pathway is basically, if you break down, are broken into 4 different parts. And you narrow it down, the funnel gets narrow. That means the holistic picture will only be clear when you know all these 4 aspects of an organism. Until you know all of them, you don't know what is happening, because one gene cannot change everything. One RNA cannot do everything. One protein cannot do everything. One metabolite cannot do everything. So the holistic picture, if you want to understand, you want to really understand what is happening. Then came the questions. When you talk about DNA, that's the genomics. So we hypothesize what can happen. So I'm looking at not at one particular gene, but I'm talking about a genome. But in transcriptomics, when you talk about RNA as a whole, so we say what appears to be happening, not necessarily happening. So proteins are the culprits who are making the things happen. So we talk about proteomics. But the real person which is working on that is the metabolomics. So we talk about metabolism of the individual, and through metabolism, we're talking about pathways. So this is what is exactly translating into phenotypes. And this is how the sequence of analysis became. So now you can see bioinformatics, when we're talking about one gene, now it has taken a shape of holistic workout, a holistic view of understanding what is happening. So it has become this. If you talk about what is happening, you talk about genome, we talk about epigenome. So you can see the databases is listed over there. TARE, Arabidopsis, RRTF, Egris, DATF, Signal Database, EPIC website. These all are gene families and genes and genomes. And all those informations are lying over there. That's all about genome. But when genome is getting into transcriptome, there's another set of databases or another set of information start coming up. That is AT Gene Express, Gene Investigator, Arabidopsis, MPSS, and all those. So this became the basis of understanding what is happening, whether my gene is expressing and it is making mRNA or not. And if mRNA is there, what is the quantity of that mRNA? Then mRNA started converting into proteome. This proteome, this becomes your proteome database. So the mRNA which is there, whether it is translating into protein or not, maybe it has got degraded in between, something like that happened. So proteome became another science. So subcellular localization, interactome, all these terms started coming up. And then protein, when they participate in some kind of metabolic pathway, it leads to the formation of metabolites. So metabolic map, metabolic profiles, hormone profiles, the pathway databases like Aracyc, Reactome, RiceFoxDB, Gall metabolome database, all those started coming up. And finally, what we are looking at, we are not looking at the molecular aspect of it, but what we are looking at, what exactly it looks like. That's phenome. So phenome is the integration of all the information and finally say, okay, this structure or this leaf, this foliage or this leaf is yellow only because this metabolite is there. This metabolite is there because this protein is there. This protein is there because this mRNA is there. This mRNA because this gene is there. And behind every field, the backbone is of bioinformatics. So you can see bioinformatics is now no more a database, no more a sequence, no more a structure, and no more a pathway. It's everything, starting from gene to the phenome. So everything, so every line became a database, it became an analysis module, it became an information. Everything becomes at the bioinformatics. So at the core, bioinformatics solves many things. So we talk about various fields of it. Genomics. Genomics is basically the study of whole genome of organism. So genome, as you know, entire genetic makeup. When we talk about entire genetic makeup, so there are multiple areas which comes up in genomics. One is functional genomics. When we talk about functional genomics, we talk about which gene is expressing and in what condition. When we talk about comparative genomics, I'll show you one of my own research in comparative genomics, where we start comparing the different genomes and see which gene is expressing and which are not. And third came the structural genomics. That means the information about their own genomes. And what kind of information? I'll show you some ideas about it. So this became three different arenas of genomics. So if you see this diagram published in 2011 in Plant Cell Physiology, you see one gene at the corner is defining the whole gamut of exercise. When we talk about only one gene, not every gene, one gene will talk about compounds, will talk about its protein, will talk about its structure, will talk about its transcript, will talk about its genome sequence, and what other. And then finally, we talk about its profiles, metabolic profiles, subcellular localization, bioimagery, expression, all this kind of information. And then finally, what kind of pathway, what kind of interaction, what kind of things, what are the co-expression networks, and finally at the phenome level, natural variation, mapping population, and all those things. So this I'm talking about one gene. Forget about talking about many genes, millions of genes. So if you take one organism and you take one gene, you can think what kind of information it can make around you. So bioinformatics becomes a complex subject. So we have everything. One gene can give you so many information, and those information, how to analyze, how to think, becomes the aspect of bioinformatics. So this is an example. When you talk about genome, when I say about genome, so you can see the type of information this genome can take. So when we talk about this genome, you can see the genome, chromatin epigenome, DNA methylome, polymorphism, insertions, mutation sequences, cDNA, homology-based relation, phosphorylation, gene annotation. I am talking about a single genome. Even I'm not talking about multiple genomes. So one genome will have so many informations, and all those information can be a different database in itself, can be a different knowledge in itself, can be a different analysis in itself. So you can see the variety of information one genome can give you. And as I have shown you, the type of information a gene can give you. So when we talk about transcriptomics, it's all about RNAs, right? But RNA is not of one type, so multiple types. The type of mRNA and the type of role they play is totally different. So it becomes a field in its arena that what kind of thing I'm looking at. Because tRNA has some other role, mRNA has some other role, rRNA has some other role. So that means the flurry of RNA which is being made are being consumed in various biological processes. And those biological processes, when investigated at various levels, can give you an idea how the physiology might change. So this is what it's all about, letting the information through transcriptomics. But it's even become a complex arena. So you talk about isolating RNAs. So single cell RNA sequences, which is a very famous concept in the infectious disease biology now, because when we talk about a tissue, we talk about multiple cells which is present in the tissue, but there are multiple cells. They are not one cell. They behave differently. So the technology which people have started using is the single cell technology, where they make 3D cultures of single cell of the tissue, and then they analyze what it is behaving and how it is behaving and how it is responding to various conditions. So that becomes a totally different single cell RNA-seq becomes an arena, that what kind of transcription profile is happening in one cell or in another cell. I'll give you one example. When you take any plant and it's present in your field, you just change a condition. You give a saline condition, or you give a heat, or you give a wind, or a mechanical engineering, whatever you think of. So what do you think the kind of change it is happening? The first instance is the plant gets under the shock, and that shock is overcome by its pathway. So it reorients its whole programming and redo for its own survival. So that means the cell which is present at the periphery would behave absolutely different from the cell which is present very deep inside the tissue. So there are different profiles. So we talk about single cell RNA-seq. So we talk about differential expressions. We talk about transcriptome profile of each and every cell at different location of the tissue. So those things, when get inculcated, it derives a different kind of information. So this is how it is done. So these are the general tools which is done for the transcriptomics. So ABC and is ARTH sequences, CDR, fine splice, TIGAR, these are all different tools for the quantification of gene expression. When I say quantification of gene expression, that means gene is expressing, but there are different copy numbers of mRNA which is happening. And more the mRNA, that means the type of signal is being stressed at specific instance. So we know which pathway, which gene is working most in certain conditions. So these quantification of mRNA will give you an idea that which gene is expressing at what level. So different stress would have different level. Different conditions would give you a different level of transcriptome. So transcriptome gives the exact snapshot of what is happening to the plant and what kind of reprogramming is done by the plant for its own survival. So there comes the proteomics. When mRNA is being made, when we talk about proteome, that means the whole set. And the type of proteomes we have, interaction proteomics and expression proteomics. When you talk about interaction proteomics, there is a whole gamut of protein which gets into protein-protein interaction mode for signaling purpose. And there is a level of protein which actually helps in the expression of certain genes. So which type of protein is made more frequent gives you an idea, because signaling is behaved through protein-protein interaction. And when the signal is passed on to some gene to get expressed, then expression level takes over. So this reprogramming can only happen by the cohesive exercise of this interaction proteomics and expression proteomics. And this is how we derive the conclusion that how the overall physiology of a plant is working through. So exactly how it works. When we talk about that, how do you do the proteomics? You do chromatography, you do ELISA, you do all those sequences, and then quantification, and then finally reach the structural analysis. So bioinformatics analysis comes at the end. So that bioinformatics analysis will give you an idea that what your gene or what your protein is exactly it's all about. If it is behaving in certain fashion, what kind of structural questions or structural changes which is leading to this happen. You must have talked about or heard about promiscuous proteins. And these promiscuous proteins not only do one type of functions, but they also perform some other type of functions. If you look at some of my papers, we have done a research on very old and very new type of proteins. And when we say old type of protein, these are the prokaryotic proteins, which is present inside the eukaryotic genomes in terms of mitochondria, in terms of chloroplast, or as a genome insertions. And I have shown in that paper that the genes which were of old type still preserve their signature, but they have changed their function. And that change of function can only be happening when there is structural variation inside the protein. And how I have reached this conclusion. I have reached with this conclusion only when I started analyzing proteins not only at the structural level, but at the sequence level as well. So I am still an old school bioinformatician, which is looking at what's happening. But the question which has been done is through different framework. And the framework is not looking at one gene or one protein, but as a holistic exercise of whole genome. And this is why bioinformatics has become a complete interdisciplinary complex science, because you have started talking about high throughput data. And when we say high throughput data, this is about whole organism and not about one gene or one tissue. So proteomic techniques, so if you say there are conventional techniques, there are advanced techniques, there are quantitative techniques, which is of use currently now, and high throughput techniques to analyze a deeper insight in each and every protein. And finally, when it comes to analyzing every result, it comes to bioinformatics analysis. And this is how bioinformaticians are not only at the genome level, which I showed you that genome has different kind of information. Now the protein also has got different kind of information. Even the transcriptome has got different kind of information. So all those information at the end, when it is produced, would only be analyzed by a bioinformatics person. So the type of application which proteomics gives you and what kind of research you can look into proteomics is that if you do talk about post-translational modification, you talk about protein-protein interaction, you talk about structural proteomics, we talk about functional proteomics, we talk about mining, we talk about expression profiling, you'll find completely gamut of information at every end. So that's why I started at the beginning, the right question. A right question will give you what kind of information and what kind of thing I need to fetch from various arenas. So if you are wrong at your question, you might be leading to wrong conclusions and are therefore a wrong kind of analysis. So the question becomes more important. And this is how the proteomics work out. And let's talk about metabolomics. Metabolomics, as it terms told, that there it's all about metabolites. And those metabolites are looking at various aspects. We're looking at different fields, different ways of looking at metabolites. and the functional genomics, nutrigenomics, toxicology, assessment, or whatever you talk about, everything reaches at the metabolic end. Even if you use pesticide, weedicide, or whatever, that's a stress for plant. And what kind of metabolic profile gets changed, you need to understand through metabolomics. And this is what exactly all about. And this is done using high throughput techniques like mass spectroscopy, NMR. And these are all used to define the metabolic profile of the organism. So when we talk about future trends in metabolomics, we talk about databases, we talk about validation, we talk about automation, we talk about large sampling. So everything, if you talk about over here, who does it? It is only and only bioinformatician does it. Metabolic analysis of the data is being produced through high throughput machines. But what to do with that data and how to make meaning of such kind of data can only be given by the bioinformatics person. No other person can give it. Because you are the perfect bridge between an hypothesis and a data and an analysis. Because you take the data, you analyze the data, and you prove that whether this hypothesis is correct or not. But now becomes the major question. What is that question? Question is, you have genomics, you have proteomics, you have transcriptomics, you have metabolomics. But when I say it's holistic, then holistic means everything. And if you say all data are, as I have shown you, is a complex, different types of data. But how you integrate them? Until you integrate them, you cannot have holistic picture of metabolism or the system. So devising a methodology to integrate all the omics data, that becomes the crucial part of bioinformatics exercise in the current days. You have your own network, you do transcriptomics, make a set of networks. Let's say it's a protein-protein interaction, you have a network, something connects with something. You do proteomics, you know which kind of protein interacts. You do transcriptomics, you know what kind of interactions and what overexpression can do. So do you have different expression profile and a connectivity profile? So you make a network. Network means connecting one dot to another dot. And this dot, when get integrated, completes one particular pathway. And that pathway you will integrate using the metabolomics. That means the metabolic profile of the individual. Then you try to simulate and find out whether the phenotype which we are looking at is exactly because of which pathway, and whether the pathway which I am assessing is correct or not. And this is how you integrate and make sense of such kind of omics data. So if you look at this particular chart, you will realize how tough it has become when you talk about genomics. It starts very small arena of information. Then transcriptomics increases its arena. Proteomics, again, metabolomics, then comes to protein-DNA interaction, then protein-protein interaction. Then comes the pathways, and pathways, what is happening in that pathway, that can only be told through fluxomics. Now, what is fluxomics? Let's say A is converting to B, and simultaneously A is converting to C. But whether the preferred pathway is A to B or A to C. So flux is traveling where and what kind of metabolite? That means the catalyst, the enzymes, which has been participating in those kind of reactions, is favoring which type of reaction. That's the reprogramming of the whole metabolic pathway. And finally, because of these changes in the metabolic pathway, your phenotype is visible, which is nothing more than a phenomics. And this is how you address any particular problem in a different way. So bioinformatician is not restricted towards one particular gene. It has to analyze a holistic view. It's like writing a mega book. So you define and you think, you may go reverse, forth, back and forth, and this is how you do it. And how you integrate that particular data. There are three different approaches how you integrate that kind of data. First is the elemental approach, where you think that you are a mathematics person, you are a computational guy, and you feel that every data point produced by the biologics are the numbers, and you try to make sense of that particular data and reach to some kind of conclusion and then join this particular information. The second is pathway-based approach. That means you let every information mapped onto the pathways and then try to correlate those pathways, and then you integrate the information and make sense out of it. And third approach is the mathematical-based approach, where you try to simulate the kind of pathway which you have made and understand whether the phenotype which is visible to me, it's because of this metabolic pathway or not. So this is the three different approaches of omics data integration and making sense of it. So again, I say, who all does all those things? Bioinformatician, because you are the master of maths, you're the master of computer science, you're the master of statistics, you analyze, you know biology very, very well. So definitely you are the only person who can do it. Because if you talk about any computational person, they don't know biology. When you talk about mathematics person, you don't know biology, but the core of every analysis becomes biology. So the analysis which you put in, the analysis which you think of, becomes the subject of your research and the hypothesis. So these are the various data repositories of omics, which I say. And in terms of plants, you can find out all those. For genomics, GOLD database is there. Transcriptomics, GEO, that's NCBI. So it's there. Stanford microarray database gives you the expression profiles. That means the mRNA expression profiles. Proteomics database, OPD, Open Proteomic Database, mass spectroscopy based proteomics data is there. World 2D page is there. Lipidomics, lipid metabolic pathways. There is one more field, localizomics. That means which gene reaching to which compartment of the cell. This is called as localizomics. That means every cell is behaving in a different fashion. That means some proteins are expressed in cytoplasm, some are in nucleus, some are on membrane. That becomes your localizomics. Then you talk about interactions, protein DNA interactions. You have a bind database, you have an ENCODE database. Then protein-protein interaction, you have a string database, a DIP database, MIP database. And when you talk about functional states, the database is RNAi database, GRID database, ASAP database, where you join this information and try to find out, okay, fine, I've got certain condition. Now look at the phenomics database, whether the condition which I'm looking at is exactly sufficing what exactly I'm looking at or not. And this is how you devise the whole study. And these are some of the softwares which does the omics data integration. Cell Designer, Cytoscape, these are very famous and softwares. Even the TopNet, these are the old databases. And now you can see accessibility, open source. And when I say open source, at the first instance, when I started my talk, I talked about a Unix system. So if you're sitting on Windows, I'm sorry to say, you are on terrible position. You have to learn how to work on Unix systems where they work on open, non-commercial softwares. Almost all the softwares in bioinformatics are open source. Open source means their code is available. So if you know coding, you can modify the code and make your own software. That's open source. So they are free of cost, academic license, they're absolutely free. You can use it, do your analysis, publish it. And this is how it is done. So that's why it is written open source, open source, open source, open source, open source. So this is how you can exploit your own capability of doing science or doing computational exercise in doing such kind of exercise. So bioinformatics then and bioinformatics now, this is the figure. So bioinformatics in current state is omics, modern biology, where computational, mathematical, analytical biology is there. Then it comes to immunoinformatics, that is itself for an infectious disease, but plant immune system itself becomes one of the biggest arena now. Then network pharmacology. This is all about doing networks. So it's not about pharmacology, but it would act like a protein-protein interaction as well. Then systems biology. You analyze all those information, put them in network, simulate the whole network, find out what's happening. So you have to learn the language of computers to understand this kind of language. There are languages which we use as BioPax, SBML, PSIMI, CellML. These are all languages which you have to understand, do some kind of simulations. And this is how you can integrate all the informations. So the question is, this science of complexity, who is the culprit? And who has done it? The culprit is sequencing. There are three basic evolutions which has taken place in sequencing era. One was the shotgun sequencing, everybody knows. Second evolution, high throughput sequencing, 454 sequencing came up. And the third revolution is single molecule sequencing. At very basic end, you can have the privilege of sequencing each and every protein, each and every gene, each and every RNA. So that means nothing is been hidden. So it's up to you, and it's up to the experimental person who talks about hypothesis, what they have to look in for. You cannot leave at the discretion of someone else to do that for you. So this is the status. You can see the type of data, gene data expansion for analysis. When we talk about petabytes, so you know GBs is gigabytes. Then after GB, it becomes terabytes. Terabytes becomes your petabytes. So that means we are increasing the level of memory. So that means the type of data we are accumulating every day is growing, growing, and growing. That means we are requiring modules of growth as much as we can. We cannot rely on one single say of technology to do that. And who is going to do that? Definitely a bioinformatics person. Biologists cannot do it. Even a computational person who doesn't understand biology cannot do it. Who cannot do it? Bioinformatics. They know computation, they know biology, everything they know. So this is how you define yourself. So if you look at different stages of genomic workflow, sample preparation, sequencing, data processing, and finally the downstream analysis. And this is how become the data management. And who does the data management? Bioinformatics. So the guy is empowered to do everything. If you see early 2010, the emphasis was on sequencing. And till today, 2016, the emphasis on sequencing started, narrowing, and data processing and analysis things became more, but the downstream analysis, storage, and understanding making the meaning of the data, that becomes huge. And if we say future, we say that the downstream analysis, data analysis, storage, differential expression, making a sense out of the data became the huge part of correct. Making data is not anything, but making sense of the data becomes everything. So this is what is the next stage. So be prepared. So when we talk about NGS sequencing, who's going to do it? I don't think biologists is going to do it. So who's going to do it? So the person who knows the software well, who knows the computation part well. So when you have a short read, you talk about epigenome, we talk about genome, we talk about transcriptome. You do alignment of those genomes, construct a genome, or using the transcriptome, or you do a transcriptome assembly. Then you ask questions. What it's all about: variant calling, abundance information, alignment free abundance, and all those kinds of things. So what's happening? So it's all about talking to the sequencing platform, and only the person who knows programming, operational softwares can only be able to do it. So this is the whole pipeline. We talk about systems biology. That's data-driven biology. And then model-driven science. And then model evaluation and refinement. So it's circular journey. So if you look at, we start from systems biology, put a hypothesis first. Produce the kind of data, we'll go to cell biology. Cell biology produce some data, go back to systems biology. And this circles goes on. And how do we do it? We talk about identification of gaps and inconsistencies. So top-down model interference. That means we had some genome, we had some transcriptome, we had proteomes, we had metabolomes. So let's look at what is missing. And whatever is missing goes back to cell biology. do this, please make the data. So they will tell you, okay, fine, this gene is expressed in this condition, goes back to systems biology. They model it again, because what they're modeling, the type of phenotype which people have been looking at. Now this is what they're modeling. And who is modeling? System biologist. And who is system biologist? Is a bioinformatics person. So therefore, the back and forth can only be done by a bioinformatics person itself. No other person can do it. So this is one example. When we talk about genome scale models, we talk about metabolic genomes. So metabolic genomes, it's all about, they are reconstruction studies. But what is a reconstruction study? That means you know everything. You construct metabolic pathways. When you construct metabolic, what type of analysis is been done till date on E. coli? Metabolic engineering has been done. Model-driven discovery is done. Prediction of cellular phenotypes, analysis of biological networks, evolutionary processes, interspecies interaction. All these being valued, understood, defined only because genome scale networks were available. And this is how you portray, and when you talk about eukaryotic system, you can think of the complexities. It's very, very complex. In E. coli, when the system is prokaryotic, it's fairly easy to understand all these complexities. When we talk about plants, it becomes very tough. That's why people are still struggling with. So this is one example which I am showing over here in comparative genomics. My research was to understand there were 3 different types of experimental data which came out of experiments. There were some expression mRNAs which were found in early response, which were found in mid response, and which were found in late response of salinity stress. So the question became, where are these genes? So we mapped them onto the chromosome using their localization, doing a lot of blast and a lot of those exercise. When the position became obvious, we plotted them and understood, okay, this is almost present everywhere. It's not about one gene or one segment of gene which is happening here. So we started characterizing which chromosome has got maximum genes. And as you know, rice plant has got different type of genes, which is bigger, smaller, everything. And these all told 12 chromosomes have different type of genes as well. So we talk about whether they are concerned towards QTLs, that means quantitative trait loci, or it's present everywhere. So what we could see over here is they are everywhere. We published this research in functional integrated genomics. And when I was requested that, tell me what functions they perform, to my amazement, a lot of genes which I don't know, they were unclassified. And when they are unclassified, that means we don't know what these genes are doing, but they are expressed in the salinity stress condition. When they're expressed, what type of things they are doing? So the question became, so that's why I was focusing on, the question became obvious. What is the function? So, and how can we solve it? And we came to the conclusion only the comparative genomics can work it. Comparative genomics will tell you by comparing 2 different organisms, working it out whether what kind of function this gene is performing in this organism, whether it has got high level of homology or similarity. We look at and assign the functions to the other gene. So comparative genomics, let's look at that. So when we did our whole genome analysis in 2 component system, we found out there are some genes and there are some proteins. So you can look at genes are 14, but proteins are 22. Genes are 16, but proteins are 19. How this change is happening? This only because gene is one, but their alternative splice products are different. That means the number of gene which is expressing is giving rise to various different numbers of proteins. And this is how we analyzed that the different pathways which is working in that scenario might be producing a lot of that kind of data. So this is, we are talking about one specific family, not as a whole family. So we recognized that there is one family called cystathionine beta synthase, which was classified as an unknown protein in plants. We annotated that by using comparative genomics, and we let's look at what this is doing, because this is unclassified protein in the plants. So I started working on that, and to my amazement, and this paper was published in BMC Genomics, where it was seen that in mammals, homocysteine is converted to methionine, homocysteine is converted to cystathionine and cysteine in mammals. And there is the enzyme called as cystathionine beta synthase. But in plants, they cannot have cysteine because cysteine is toxic to plants. So the organism has reversed the pathway to convert cysteine to methionine. And the enzyme which was cystathionine beta synthase completely got eliminated and became cystathionine gamma synthase, which actually favoring the cysteine conversion to cystathionine, and beta domain is transformed into gamma domain. But this protein is gone, but its signature is still there. So our question was what it is doing. So we started looking at, let's look at whether in which type of genes and what kind of domain it is present. So we could found that these domains are the supportive domains of the structural moiety. And these proteins become different type of function. They assume different types of function. Voltage gate, CLC, and all these are the anti-transporter workouts which they've been doing. And this is how this type of protein family is doing different types of function, may not be the same kind of function which were available in mammals. So what we found? We found that these are the domains present, but these are not the proteins. Domains are the supportive domains, and they might not have kind of functions which we are looking at. So definitely this protein has evolved into something else. And this was our conclusion using comparative genomics. In exactly the same workout, 29 genes giving rise to 46 proteins, while in Arabidopsis they are similar. Why it is happening? Oryza sativa, rice as we know, is undergoing selective selection pressure, where we have different types of genes moving into some kind of special kind of activity. If you read my paper, Old or New Prokaryotic Genes in Frontiers of Plant Sciences, you will realize that these functions got deviated, but their structures remains the same. Their sequence remains the same. And this is how they assumed new functions every time during the time of evolution. So these are the different types of functions they were performing when we analyzed the ontology. Now, when you did comparative genomics, you did omics. Now what? Now the answer of this what is the prediction. And how you do the prediction? How you recommend that if the plant is having such kind of condition, and if we change certain pathway or gene, certain new phenotype will evolve and the plant would be able to survive. How would we do that? And this is where your machine learning and artificial intelligence came. What is artificial intelligence? It's all about deep learning. That means you take a lot of data which is coming from omics and your comparative genomics. You feed that information to train your machine or your program to learn that kind of phenotype. And then you predict that if I change this particular gene, this kind of phenotype would be available. And how this is done, you can see through this slide, artificial neural network. So at the very start, these are the conditions. Okay. If you modify certain conditions, what kind of genes get perturbed? This is the middle layer of expression. And finally, what kind of processes which would be the outcome? This becomes your outcome of that neural network platform. So how do we do that learning? We take the genome sequences, we identify the changes, we learn from that. We learn means not we. The machine learns all those patterns. Those patterns are then used to predict. And when it predicts perfectly, it perfects the exact epochs line where we exactly know what kind of learning and prediction it is making. So the prediction, validation, testing, the training, validation, testing goes on until we have a perfect model to judge what exactly is happening. We predict what kind of mutation would lead to what kind of phenotypes. And this is how we do the prediction. And all this started coming through genomes and the type of gene sequences. And this is how, even if you know about this coronavirus, the artificial intelligence method have been used, but they failed to do any kind of workouts in this case. Reason being that the virus is smarter than us. It learns much better, much quicker than the human beings or the computational machines. Computational is know what is being there is happening in the system, but virus does, which nobody anticipates. That's why we are not able to catch them. But the method which we use to learn what would happen, it may be happening that after some 15, 20 years, we'll be having that what viruses might be doing, might be knowing that. And this is how we do the prediction analysis, by identifying those SNPs, let the machine learn, and then we predict. And this is how agriculture gets benefited. That if you know the drone technology which has been used in various farms, we catch the picture, take the image, use the machine learning and artificial intelligence approach to catch the changes which is happening at the foliage. We predict which plant is having the disease, or they are getting dehydrated, or they are getting some stress. So in a bunch of crop which is standing in the field, you can do swiping analysis at every crop. So you should know artificial intelligence and a biology to understand the nuances of this analysis, and thereby do a perfect prediction model who can tell that if you change this or you change this particular condition, the crop gets improved. So the overall phenotypic system which is assessed over here is something like, at the development phase, you do some kind of machine learning through infrared imaging. You predict the kind of change, and then tell the farmer that if you do in the cropping in this condition, this soil type, this much water, this much temperature, this much care, this much pesticide, you can predict the level of crop in the fields, or the type of crop in the field. So in silico biology holds an important role in plant stress interaction. Everybody knows the number of unsolved problem in bioinformatics exists. So somebody says, "Ab kya karun? Ye to khatam ho gaya. Sab log kar liya. Sabne sab kuch kar liya." No, there is always a scope in research, and there's always an unsolved question, because biology is still very far away from getting solved. So we revolutionize the way we do experiments. Now it's not about sitting on a table doing some gene or protein. It's all about taking the holistic approach from the table to the computers. So this holistic approach will solve many things, will revolutionize. So omics data motivates the researcher to create better, more integrated and easily accessible pathway databases. So we always fill in gaps using the omics analysis. So the amount of information which we get is organized, detected, integrating them, and therefore revolutionize the agricultural practices and production technologies. So I always say, biology, computer science, mathematics, information technology, everything is needed to a bioinformatician. Something to more access, something to less. If you are more analytics, then you are biologist. If you do tools, you are computer science. If you do assistive technologies, mathematics and information technologies. So that means all those things comes naturally to bioinformatics person. So you have to be a programmer, you have to be a biologist, you have to be an IT person, you have to be a statistician, and even sometimes you need to be a graphic designer, because you need to predict the pathways and a phenotype. So therefore, you need every skill in one person, but that's not happen. That's not going to happen. So how do you do it? It's all about being in collaborative work. So you talk about a lot of people getting into team, solve a particular question. It's all about making some kind of research possible. So you cannot do research in silos, but you work in team to solve different kind of problems. So most basic, use web-based tools. That's primary need of biology. Then use Unix-based tools. then you write some sort of Perl programming and do shell script to, you have got a lot of data, fetch out some data, get some meaning out of it. Then high throughput data, you do a storage, you scalability, and you devise algorithm. And the computer science focus, sitting with the biologist and asking a question, okay, what kind of algorithm you require? What kind of output you require? So I'll devise one algorithm with respect to that. And then finally, you make all these things public using databases and web pages. This is how you do. And finally, when I say advances in experimental biology can give different types of data for analysis. So you require different kind of expertise to deal such kind of data. Then your wisdom with biology comes in. You predict the computational models, you try incorporate multiple information, which becomes more complex. And finally, you use computer technology to deal with such amount of data in reasonable timeframes and make conclusions. So this is what we do. Sample preparation, sequencing, some other will do. Raw data, some other will do. But we'll do stuffs. We'll do something and make sense out of it. And this is what the new bioinformatician is all about. So thank you very much. And I'm open for